Thank you for joining me today. My name is Alicia Henn from Biospherics, and today we have a very exciting interview with three cytocentric visionaries from the King Abdullah University of Science and Technology. We have Samhan Al-Salami, we have Dr. Shannon Klein, and Professor Mo Lee, who are part of a larger group of cytocentric visionaries that have published 3 landmark papers in the last few months. And check out the titles on these papers. First one's called A Prevalent Neglect of Environmental Control in Mammalian Cell Culture Calls for Best Practices. This was published in Nature Biomedical Engineering. The second paper is in Nature Communications Biology, and it's called In Situ Monitoring Reveals Cellular Environmental Instabilities in Human Pluripotent Stem Cell Culture. And a third in Frontiers and in Cellular and Developmental Biology called Toward Best Practices for Controlling Mammalian Cell Culture Environments. And in this paper, they've outlined a set of guidelines called the PRINCE guidelines, or the Preferred Reporting Items Describing the Nature of Cell Culturing Environments, which sets out a very easy list for researchers to follow in order to actually report the conditions for their cells and tissues and culture in such a way that someone else can pick it up and actually reproduce those conditions in their own lab. So all of these full citations of these papers will be at the end of the interview. It should be required reading. I'm going to stop talking now and get to the interview because I think you're really going to enjoy this. It was a lot of fun talking with these cytocentric visionaries. Can I start sort of at the beginning? What made you want to write these papers in the first place?
First, thank you, Alicia, for inviting us to have this conversation. I'm a stem cell biologist, so I know stem cell culture can be very finicky, and environmental factors like pH and oxygen and gases are known to be important for stem cell self-renewal and differentiation. However, very little is known about these factors in actual culture. And there are also known variability in stem cell differentiation, and that pose a big challenge to research in this field. So I'm very interested in improving stem cell culture and optimizing it by doing more reporting, monitoring, and controlling of environmental factors. But as a stem cell biologist, I lack the expertise to really monitor and manipulate these factors. That's why we formed a collaboration with our marine biology scientists who have a deeper expertise in understanding environmental factors. So that Dr. Shannon can tell us about what they are working on and why we came together.
Yeah, thank you, Mo. So as marine scientists, we place a large focus on how marine organisms interact with their environment. And my research, and with Professor Carlos Duarte's research, we have a big interest in dissolved gases and what we call carbonate chemistry in the ocean. And when we think about early metazoans, they came out of the ocean and they essentially they internalized seawater. And this is very similar to the makeup of our bodies internally. So we're actually quite good at this type of chemistry within the human body. And this is something that as marine scientists, we place a big focus on. And so with one of our first meetings with Professor Mo Lee and Samhan, one of the first things that we were doing is we were just comparing notes between what biomedical researchers do to control their environments in their cell cultures and how that really compared to what we were doing. And this was really some really exciting meetings for us because, you know, the differences were quite astounding and discussing methodology, but also the challenges for how our methodology has to be adapted to work in these, you know, sterile and human cell environments. So that's really how it got started.
Wow, the idea that cells and culture are related to marine life is kind of mind-blowing when I think about it. When I think about cells in a liquid culture, like in a static plastic flask, like in a T75, that's a little ocean there for them. And that's kind of a neat idea. I could see why that would be exciting. That's exciting to me too.
Yeah, exactly. Yeah, this is a fascinating start.
Yeah. Yeah, that's really cool. Now I'm thinking about like macrophage as like octopus, and like, you know, different cells as like different sea creatures, and the medium that we provide them as like the ocean. It would be an ocean from their point of view. So what are the key points in these papers that you want everyone to remember?
I would like everyone to remember that the current cell culture practices in biomedical field are inappropriate to allow for research reproducibility and the utility of scientific findings. Indeed, we found that recently in our work that as cells grow in this tiny ocean or tiny media, they act to, over time, as they grow, they act to deoxygenate and acidify the media. And this phenomena has been overlooked by cell biologists. And so there is an urgent need not only to monitor, but also to control and report the values and set point of these factors.
When you really start to sit down and you think about all of the variables that can affect dissolved gases and acid-base chemistry, you can write a really, really long list, like pressure, salinity, osmolarity. All of these things will affect environmental parameters. And when you think about all of this potential variation, it starts to become really noticeable that we really need to start measuring this and paying attention to it. And in some of the results that we've found, like almost all of the cell cultures that our group has monitored and other research groups have monitored have seen, you know, substantial instability. And, you know, this is partly due to variation between flasks, but also among different types of cells.
Mm-hmm. So what do you say to researchers that think that, oh, there's too many factors for me to try to control them all? I'm just going to give up and do it like I've always been taught.
Yeah, so I think I mean, in biomedical science, you know, they're spending a lot of money and a lot of effort measuring some amazing, you know, cellular responses, the cell identity, and there's a lot of money and effort into looking at these responses. But these responses are going to be determined by the environment in which they are. And so if these responses are a direct result of conditions within a flask, then I think it's vital that we understand that environment. That's a really, you know, basic thing. And, you know, by measuring it, we can then start to resolve it. I think that at this stage in the field, we first need to identify what's going on and then start to improve conditions. I think we all believe that this is not something that's going to happen overnight, but there's going to be, you know, smaller steps to start fixing the problem. And as, you know, new technologies and more affordable systems come on the market, it will become a lot easier. So it may seem really overwhelming initially, but once you kind of set up that infrastructure and you start kind of chipping away at this task, it starts to become easier. And I think, you know, in our research collaboration, we had, you know, it wasn't easy for us to do these measurements, and it took us time to solve problems. But now that we have identified what the best things are and we're sharing that with the research community, it starts to become more manageable, and things that would have taken us weeks to figure out, now we can do within 10 minutes, you know?
Yeah, definitely. Just to follow with Dr. Shannon, what other researchers need to realize is that these cells are not static, they grow. And although people could, or scientists could buy commercial media that supposedly have the right pH, this pH change over time. And these changes, these factors start to interfere with experimental findings. So the result that could be found in one lab could not be probably reproduced in another lab. So for that, it's very critical that we need to monitor these factors and report them.
Yeah, so I just want to add 2 points. First, for people who think there are too many things I need to keep track of, and I rather not spend the energy and time to do it, think about how many replicates you have to do. If you don't know the extent of variability in your system, and that's added cost and added efforts. If you carefully monitor and control the environment, your conclusion and data and conclusion could be much more strengthened. So this actually may save you time and give you better results. So that's one point. Another point is we are trying. So by first looking at the in the literature, looking at the practice of reporting and controlling environmental changes, and then we also did actual measurement in stem cell cultures, and then we try to provide some practical guidelines for researchers who want to pay attention, want to do better. And that's our recent paper published in Frontiers of Cell and Developmental Biology. And hopefully this is a starting point to change practice in cell culture and help everyone to obtain more solid and more robust results.
When we know that there's this kind of linear drift in conditions, you can use even standard pH and oxygen sensors, which are really easy to get and probably already available in most labs, to measure at least the extent of before and after in this type of variation. And then there's a number of things that you can do to minimize that, just through the knowledge of how metabolism works and cell density influencing how extreme these changes might be. So one of the things that we've discussed is reducing cell density, potentially incorporating more medium exchange, increasing the header space. So, the ratio between cells and then the header space in the flask that they're exposed to, that will quicken the equilibration between the atmosphere and the medium. And so there's a number of factors that you can do right now that can help try and minimize this drift into within, an acceptable range of variation. And that is something that we should be looking to the human body to see what kind of variation is or isn't acceptable.
What are some ways that new technology could tackle this problem of being able to report all the conditions that cells are exposed to during experiments?
Yeah, so with a lot of journals, they dictate really strict, word counts or page numbers, and often limit the amount of information that you can publish. And that can lead to, unanswered questions about what the method actually entails. But there's a lot of things that are available. For example, most publishers will allow you to add extra information on a method in supplementary information. And I don't think I've ever published in a journal where there's been a strict limit on that information. There's also a number of online repositories that are available and are open access to everyone around the world. So I know that with some of our studies, we've always made our methods publicly available, and we've made all of the information that we can think of that could be useful available. There's also a number of I think Nature Protocol Exchange is also a really good initiative where you can publish your protocol and openly exchange it with people around the world. So I think, as much as there are page limits and word count restrictions from publishers, I think there is numerous avenues to make your method and your protocols publicly available and share them with the community.
There are other journal where you Jopi, you can take video of your experiment. So, and you publish a video protocol that basically show the community the know-how you do the experiment. This allows for enhanced reproducibility.
Yeah, there are new types of data generated in the experiment we have been doing. I'm sure Biospheric's equipment generates log files, right, for experiment. These are very valuable data. And in our experiments, we sometimes use bioreactors. We generate huge log files of all the conditions the cells experience. So these should be part of the publication, at least for the typical experiments.
Something that publishers could do is use these Prince guidelines to change the way that information is reported in their journals. What is something that the funding agencies could do to support improving reproducibility this way?
Yeah, so we a number of our recent papers that, you know, some of the challenges are the lack of infrastructure and the affordability, because this can be quite expensive to integrate logging systems, especially for dissolved gases like carbon dioxide, which are critical for cells and acid-base stability, but they're, you know, notoriously difficult to measure. And so, you know, this is a challenge. So funding agencies should certainly encourage research initiatives to not only improve the technology that's available to labs around the world, but also to explore the effects of this instability. Hopefully it's not just about improving conditions, but also going to drive some really new and interesting questions. And I know just from our collaboration between marine scientists and biomedical scientists, we've already come up with, you know, some really intriguing questions that are interesting on both sides.
Mm-hmm. Do you want to tell us a little bit about what kind of new questions that have come up in actually monitoring and keeping your eyes on the cell environment?
Sam, do you want to?
Yeah, so there are very important questions, especially to the cell physiology, and how these cells behave at the molecular level, how they behave in different environments, how they respond to physiological pH, and how they also how the environment, controlled environment, influence the cellular behaviors.
Some examples can include cell identity. For example, during cellular reprogramming, pH seems to play a role in the reprogramming of somatic cells to pluripotent stem cells. And there are also known effects of low extracellular pH on cancer cell proliferation and metastasis. To really study this under controlled environment, to be able to control environmental factors tightly, should be a very important research area. So we are also interested in doing some of this work using technologies we're developing. So that will be our next paper, hopefully.
Is this going to start sort of a new wave of conditions for cells that are completely different from what we're used to in traditional cell culture?
Yeah, so in stem cell field or regenerative medicine field, there has been a huge struggle to make progeny cells, let's say blood cells or other cell types that mature enough to be used for, let's say, clinical therapies. And so there has been a lot of attempts using very expensive technique to understand the process. And it could be very well that these environmental factors is undermining the differentiation of these cells from human pluripotent stem cell to these different tissue. So making sure that the environment is well controlled to mimic the physiological niche of the specific cells of interest could very much enhance this process and could basically propel a huge advancement in biomedical research.
What do you say to people that say, I want to match my old papers with the conditions I provide for cells now? I don't want to change from what I've published already.
I think that's going to be very difficult because we know that, you know, even the slightest changes in between individual flasks will create substantial variation. So in one of our papers, we actually looked at, experiments that had measured this exact same flasks, and the variation between the flasks that are prepared exactly the same in the exact same laboratory conditions, even between them, had huge variation that we wouldn't expect. So if we're not measuring and reporting, there's not really any way that we can mimic something, especially if it's done in a completely different time or a different setting with different materials. So this is another reason why it's so vital to capture that variability and to ensure the reproducibility of the experiments.
Mm-hmm.
Yeah, in another word, to say that I'm repeating exactly the old experiment may be a wish that cannot be fulfilled, because you could never reproduce something you did not measure and document extensively.
You can never repeat something you haven't measured. That's profound. This could have a tremendous impact on the way that people do cell-based science. What do you envision that this will look like when we really have good control over cell conditions?
I think that's a really interesting question. It would be automation, it would be alerts and things like that, really nice and simple sensing technology that doesn't need these huge calibration steps, things that can be reused as well. So these new sensor spots are really fantastic, and things like this, but the more automated it is, and the more precise it is, the easier this is all going to be. And at the moment, the major challenge is the lack of this technology. And then if we do have this technology, it's just very, very expensive. And then for a lot of research institutes, just not obtainable.
I think the automation will add predictability to cell culture, especially if you create potentially a system where every time you culture a specific cell type, you would have an automatic electronic checklist. If you want to culture, let's say HIC293, you would get the system tell you need to add this media, and then there would be like type of sensor that sense the environment every minute and provide the complete log of what you added and what happened to your cell culture. This log could be sent to a different lab having the same equipment, and it could be repeated. And you very much, if you find the issue with your data, you can go back to the logs and you will potentially find the issue. And also you would know if this type of automation, you would know if you get contamination, let's say bacteria. The system will tell you there is an foreign species invading your culture, so you would get in basic notification. And so you are aware of what's happening to these cells. And so, but the current batch culture system is very much cells in the dark.
So what's the problem with using animals to control the cellular environment for us?
I think just my take on this is that mice or animal work is increasingly under a lot of ethical concerns, and there's a huge push to use cellular model like stem cell and others. That's first. Second, there are in cell culture, let's say human, let's say brain, mouse cannot, for example, cannot model that. So we are very much different. The closer thing you can get to this or to understand the brain is brain organoids. So there is a huge limitation when you want to study human tissue and health and disease. So mice cannot be used to answer all the questions. And just the fact that you are introducing it's foreign tissue that already create potentially it might not be applicable to humans. It could basically, a certain drug could work in the mice, but it might not work in humans. So there is a huge interest to improve the cell culture to a point where we could actually translate it directly. It could be mimic the human physiology, so it could be directly translated to human.
Yeah, and also so far as I know, cells cultured or cells grown in an animal host cannot be used for human therapy. So if we're talking about future human cell therapy, the cells needs to be grown under controlled, clinical proved environment. So the better we can make this environment appropriate to cells, the better we can improve the functionality of these cells.
So cell cultures are needed because they're human cells, they're not animal cells. We need, you get better translatability if you're working in human models rather than animal models. The ethical issues of using all these animals, and as well as the inability to then take cells that are grown in animals and move them into the human clinic. Those are all really compelling reasons to make human cell culture more human. Oh, one other thought I had from before. What do we say to researchers that are afraid to share, that are afraid to make things easier for other labs to reproduce in an environment that can be kind of competitive?
One thing that comes immediately to my mind is if no one can reproduce your results, who is going to cite you? So I think that's going to be a problem.
I have a motto for this, and if you're competitive with someone, collaborate.
Also makes sense.
That's only going to be better for you and them, and then you don't have the issue of keeping secrets.
Yeah, and definitely you want people to work on this topic, so you could cite them and they cite you, and you become make a science a happy place.
I like that a lot. Now, is there anything else that you'd like people to know about your work?
I would say that marine scientists and biomedical scientists work really well together.
Yeah, that's agreed.
Maybe biomedical scientists can go through their faculty now and start making connections to do some work. That would be cool.
I would like to let everyone know that the work we are doing has a broad impact on biomedical research. In this broad impact will slowly allow researchers to realize that the basic things of what make our body work is missing in cell culture, and realizing that will encourage to control these parameters and guaranteed will enhance predictability of the cell culture and enhance the translation and application of human therapeutics.
Thank you very much. I really appreciate you joining me today. And I thank you for your time and your consideration. And I look forward to taking your message and spreading it as far as we can and as loud as we can, because these issues are absolutely critical for advancing biomedical science.